Hello students, and we're going to go ahead and move on to the 6.2 notes in this video. In these notes, we're going to look at gene expression and regulation, which involves, so gene expression is the actual process of taking the information in your genes, in your DNA, and then expressing it. So actually taking that information and using it to build proteins. And then those proteins will then do millions of important things inside of your cells and inside of organisms. And it is what ultimately gives you all of your traits and characteristics. So we're going to look at how that process takes place, like taking the stuff, this information stored in your DNA, the blueprint of the cell, and actually using it to make things. And then we're going to look later on how that's regulated in cells and how cells control which genes they're using and which genes they're not using and when they're using certain genes and how much of certain genes they're using. And so we'll get to that later on. But in this video, we're going to look at topic one. In topic one, we're going to talk about protein synthesis. And so protein synthesis is gene expression. This is the process of taking a gene, which is a special sequence in your DNA that has the instructions to make something. And so in protein synthesis, what's going to happen is that we're going to take that information in a gene, which is just a sequence of nucleotides, A's and T's and G's and C's. And this will be how, during this process, we'll see how those letters, how that sequence in your DNA ultimately is used to put together amino acids, which are the building blocks of proteins. So we're going to use that information to actually put together amino acids in a specific order that will then fold up into a specific protein that has a specific function that does stuff in your cells. And so this process of protein synthesis involves two steps. There's a step called transcription and a step called translation. So we'll take a look at both of those in this video. But just to remind you guys about what a gene is. So like I said earlier, so you have a bunch of DNA made up of nucleotides, and genes are these special sequences in your DNA that actually have the instructions to make some sort of functional product. In most cases, it's a protein. It's an instruction to make a certain protein in your guys' genes. And then other cases, there's some situations where it's an RNA molecule that's being coded for in these genes, and that RNA molecule has a special function as well. But the huge majority of the time, we're looking at a gene that codes for a protein. And then that protein does something important in your cells and in the organism. And so to do that, these two processes, like I mentioned, these two steps, transcription and translation, here's the overview. Basically, what we're going to do first is transcription. And transcription, we're going to take the sequence in your DNA, which is all these different nucleotides containing adenines and thymines and cytosines and guanines. And we're going to first copy that information down onto a RNA strand. Specifically, it's going to be an mRNA strand. So we call this messenger RNA. Messenger RNA, or this piece of RNA here, is basically a copy of the gene. It's complementary to one of these DNA strands in your genes, and we're going to copy down that information, these letters here, and then we're going to take that mRNA and use it to do translation, which is the second step. In translation, we're going to take that information in the mRNA and actually use it to put together amino acids. There's 20 different amino acids. Those amino acids are the monomers that build proteins. And in the mRNA, we can actually use that information, these letters in your mRNA, to figure out what amino acids are going to be put together and in what order. That process is called translation. So in transcription, we're taking your DNA and making mRNA. And in translation, we're taking your mRNA and then using it to put together amino acids, which will become the protein. And so that flow of information is known as the central dogma of biology, or specifically molecular biology, where in life on this planet, all this information is kept in your DNA. And then the flow of information goes from your DNA first to RNA, and then from RNA to a protein. And that's true for all life on this planet. It's a very unifying characteristic of life on this planet. So let's get into it. Starting with transcription. So like I said earlier, transcription is when we're going to take the information in your DNA and then copy down that information onto a piece of mRNA. And so to do that, first of all, this process is going to take place inside the nucleus of the cell. And so your DNA is kept protected inside the nucleus, at least in eukaryotic cells. And so transcription occurs inside the nucleus. And there's an enzyme who's going to do this job of transcription, who's going to read the information in your DNA and use that information to build the mRNA, the messenger RNA. And that enzyme is RNA polymerase. So RNA polymerase is this little blob right here. He is the enzyme responsible for doing transcription. He's going to find genes on your DNA. And then once he finds a gene on your DNA, he's going to unwind the 2 strands of DNA there. And then he's going to use one of those strands to put together an mRNA transcript. So he's going to build this mRNA complementary to one of those strands in the DNA in these regions that contain genes. And so when that RNA is being built, it's basically going to be made, like I said, complementary to one of these strands that it's reading. In this case, in this picture, you can see it's the bottom strand that it's looking at, and it's making this mRNA based off that bottom strand. So this mRNA would be complementary and follow the same base pairing rules as DNA, where like G will go with C or C will go with G. The only exception will be there won't be thymine. Instead, there's going to be uracil. So instead of adenine going with thymine, once we make the mRNA, it's now going to be adenine going with uracil. So you'll see that, and we'll look at some examples in a second, some practice questions soon. But that's the gist of what's going on here. And you guys can also see that mRNA that's being made is being made anti-parallel to, it's complementary and anti-parallel to that strand that the RNA polymerase is reading. And then another thing I want to mention real quick is that this process of transcription involves, it's basically just RNA polymerase who's doing all of this. And so don't get this confused with DNA replication. During DNA replication, we were taking a DNA molecule and replicating it, which involved a huge group of enzymes, including helicase and topoisomerase and DNA polymerases and primases. Whereas in transcription, RNA polymerase can basically help himself to the DNA, and he doesn't need a bunch of other proteins to like help him unwind the DNA. We don't need helicase to do that. He actually himself, this enzyme, can unwind your DNA and read it and build the mRNA. And so he's pretty good by himself here. And so all of this starts, the spot that this starts on your DNA is a region called a promoter. A promoter is a sequence in your DNA where RNA polymerase will go and bind to. So this is basically going to be, you're always going to find a promoter right before a gene in your DNA. So you guys have a lot of DNA. There's a lot of, in all organisms, there's a lot of DNA. But what's interesting is actually less than 2% of all your DNA, you have billions and billions and billions of nucleotides of DNA, and less than 2% of all that DNA actually is genes that code for you as an organism, like code for all your proteins. It's less than 2%. The other 98% is actually what we call non-coding DNA. It doesn't code for anything. They're not genes. So a lot of your DNA is just there, and it doesn't have the instructions to make anything. Only 2% of your DNA actually have the instructions to build proteins, for example, and only 2%, less than 2% is genes. So the other 98%, we'll talk a little bit about some of that DNA later. Not to say that some of it's not important. Some of that, the other 98% of your DNA, some of it is important, but they aren't genes. And so we'll talk a little bit about that later. But anyway, the reason I brought that up is because for RNA polymerase, he needs to know, okay, where are the genes? He's not just going to randomly go find a random spot on a chromosome on a DNA molecule and just start making mRNA. He's always he's able to find genes. And the reason he's able to find genes that actually code for things is because all of your genes have a special sequence called a promoter. A promoter, again, is the sequence, a special sequence in your DNA that RNA polymerase will find, and that's where he's going to start doing transcription. So you can see here he comes, RNA polymerase, and now he's going to bind here to this sequence of DNA and start unwinding it and building the mRNA. So in all your guys' genes, you always have a promoter sequence, the special sequence, to basically flag down RNA polymerase to tell him, like, here, look, there's a gene here. This is important. Please come read this DNA. And then what you also see in this picture are these other proteins called transcription factors. A lot of times, these transcription factors bind to the promoter and they help RNA polymerase come find the promoter and bind to the promoter correctly. And so these guys are pretty important, mainly in just eukaryotic organisms. But we'll talk more about these later in a future video. But for now, just know that they exist and they basically just help this whole thing get started here with RNA polymerase. And then the other thing that's on this picture that's worth mentioning is something called a TATA box. A TATA box is just a high concentration of adenine and thymines that are repeated over and over again that are found a lot of times in promoter sequences in eukaryotic organisms. So in eukaryotic DNA, like your guys' DNA and other eukaryotic organisms, the promoters, oftentimes we find, almost all the time, these TATA boxes. And so it's just a coincidental thing that happens to be found in these promoter sequences in eukaryotic DNA. And it actually has a lot to do with helping these transcription factors find the promoter and bind there with RNA polymerase. So then once you bind to the promoter, you can get started. RNA polymerase is then going to start unwinding the DNA and building the mRNA. RNA polymerase is a lot like DNA polymerase. So DNA polymerase is the enzyme who builds the DNA. RNA polymerase, so DNA polymerase is involved in DNA replication. If you guys remember, when we replicate the DNA, he builds the new DNA strands. RNA polymerase is not building DNA, it's building RNA, the mRNA in this case. But there's a lot of similarities. He also can only read the DNA in the 3' to 5' direction. And as it's reading the DNA in the 3' to 5' direction, it's building the new mRNA in the 5' to 3' direction. So just like DNA polymerase, RNA polymerase can only build in the 5' to 3' direction, which if you look at this picture here, he's building constantly off the 3' end when he builds the mRNA. And he's doing that while reading this strand 3' to 5'. So this was the 3' end of the strand that he's reading, and he's working his way towards the 5' end. And as he's doing that, he's building this mRNA complementary and anti-parallel in the 5' to 3' direction. So keep that in mind. That's the specificity here going on during transcription with this enzyme, a lot like DNA polymerase. And so that's going to continue happening until he gets to a special sequence called a termination sequence, which will it's a special sequence of DNA that tells him, okay, you're done. You can stop transcribing and making this mRNA. And then he ends up letting go. The RNA polymerase will let go and then release the mRNA. And now you have your mRNA. So that's the gist of what's happening during transcription. Again, all this is taking place inside the nucleus of eukaryotic cells where your DNA is. And so RNA polymerase, he finds a promoter, he latches on, and then he starts reading and transcribing that DNA into mRNA. One thing to point out here is that there's 2 strands in your DNA is obviously double-stranded. And when RNA polymerase goes and finds a gene to do transcription and make the mRNA, he's only going to read one of the strands. The strand that he reads that he builds the mRNA based off of is called the template strand. Unfortunately, though, it also has other names, and so it's kind of confusing. But the strand that RNA polymerase is using to build the mRNA, it's called the template strand, which is also called the non-coding strand or the antisense strand. So all of these are synonyms. It's the non-coding strand or the template strand or the antisense strand. That's all referring to this strand right here in this picture that RNA polymerase is reading and building the mRNA complementary to. Now the other strand is called the coding strand or the sense strand or the non-template strand. That's this top strand that he's not using. Okay, and it depends on the gene. Sometimes it's the top strand, sometimes it's the bottom strand. In this case, it's the bottom strand that he's using. That's the template strand that he's making this mRNA. If you guys look, it's complementary and anti-parallel to this strand, this template strand that he's reading, or the non-coding strand. Some students ask, why is this called the coding strand and this one's called the non-coding strand, when in fact he's using the non-coding strand? It seems weird. Why wouldn't you call this the coding strand? And the reason this top strand is actually called the coding strand is because RNA polymerase is making the mRNA complementary to the template strand. It actually has the same sequence. It has the same code as the top strand, the strand that it wasn't using. which is why we call it the coding strand. The coding strand actually has the same code as the mRNA. The only difference is instead of T's, there's going to be U's because this is mRNA. mRNA doesn't have thymine, it has uracil instead. So if you guys read this top strand, it says A, T, G, G, C, A. Well, that's the same thing down here. A, U, G, G, C, A, and so on. The only difference is the T's are now U's, but it has the exact same code, and the 5' N and 3' N are exactly the same. So the coding strand actually matches your mRNA, except for the thymines becoming uracils. So that's why it's called the coding strand. But technically, that wasn't the strand that was being used by RNA polymerase. RNA polymerase was using the non-coding strand, or the template strand, to build the mRNA. We'll go over that more in class, but that's what's going on there. So after the mRNA is made, in eukaryotes, we actually modify the mRNA before it leaves the nucleus. So there's some modifications that we make to the mRNA before we let the mRNA strand leave the nucleus, because this mRNA is now going to go find a ribosome. That's what it needs to do next. The mRNA, after it's made, is going to go find a ribosome, and that's where the next step is going to take place, which is translation. Translation takes place with a ribosome. But before we get there, in eukaryotes, we're actually going to take the mRNA that we just made and we're going to modify it in a few different ways that I'm going to point out right now. And this is still in the nucleus before the mRNA is allowed to leave the nucleus. And so the mRNA that we made initially is called pre-mRNA. It's not technically ready yet to go do translation. And so this is what's going to happen. Here's how we're going to modify this before this mRNA. Before it's allowed to go do translation with a ribosome. And this, I want to emphasize again that this is only in eukaryotes. In prokaryotes, like bacteria, when they make mRNA, their mRNA is good to go. Like, they don't mess with it. Like, they make their mRNA and then they send it to a ribosome and do translation. But in eukaryotes, this is a big difference. In eukaryotic cells, we modify it in the following ways. One thing we're going to do is we're going to add something called a GTP cap to the 5' end. So we're going to hook on this little molecule piece here. This molecular piece is going to be added to the 5' end of the mRNA. And then we're going to add something called a poly-A tail to the 3' end, which is basically just a bunch of adenines repeated over and over again to the 3' end of the mRNA. So the mRNA will get a 5' cap, and we'll get a poly-A tail on the 3' end. These things do have a couple different purposes. They help with the mRNA, first of all, getting out of the nucleus. So in order to get out of the nucleus, the mRNA has to have a GTP cap and a poly-A tail. If not, then it's going to not be able to leave the nucleus. And then it also helps protect the mRNA once it's in the cytoplasm of the cell. In the cytoplasm of the cell outside the nucleus, there's a lot of different enzymes and chemical reactions taking place, a lot of things out there that could destroy or damage this. And the poly-A tail and the GTP cap help protect this mRNA, which has a special set of instructions that it just copied down. And then it also helps it find a ribosome, and it helps the ribosome attach to the 5' end. And so this is going to make it a lot easier to leave the nucleus, to be protected once it leaves the nucleus, and to go find a ribosome. That's why these things are being added. And then another thing that happens is in your guys' mRNA, in your genes, and again, this is only for eukaryotes, in eukaryotic genes, there's these random pieces of DNA within the gene that actually aren't part of the gene, and they're called introns. Introns are these random disruptions within your gene that exist that aren't part of the instructions to make something. So if I was looking at a gene in your DNA, a gene in your DNA would have the instructions to make a certain protein, but within that gene, that sequence, there's going to be these fragments, these sequences of DNA that technically are just random nonsense. Like, they're not part of the instructions. They're called introns, and there can be a lot of them sometimes in your genes, and those introns need to be removed. So it's kind of like if a gene was like the instructions to, like, let's say instructions are a recipe to like make cookies or something. Like, there's a gene that you guys have in your DNA with the instructions to make chocolate chip cookies. And in that gene, it has the instructions on how to do it. It tells you to do this and then do that and then mix this and then add the eggs or whatever. But then randomly, when you're looking at the recipe, it's like someone just like smashed a keyboard. It's just like random letters everywhere. Like it's not actually part of the instructions. And then you keep reading, and then all of a sudden it says, okay, step 5, you do this. Step 6, you do this. And then all of a sudden again, it's like someone was very angry on a keyboard, just typing a bunch of random letters that aren't the instructions for anything. And you keep going, and finally you get back to the instructions, and it says step 9 and step 10, and then put it in the oven and so on. But those random disruptions within the gene, those are called introns, and in eukaryotes we have to remove those after you make the mRNA. So when you make the mRNA and you do transcription, you're going to copy all of it, so the whole gene from beginning to end, but within that gene are these random pieces that technically aren't part of the gene. So your gene is kind of split up with these random sequences of DNA that technically aren't part of the instructions. And so once you make your mRNA, you copied all of that, and those introns you're going to have to cut out. You're going to have to remove them and put together what's called the exons. The exons are the regions in your guys' genes that actually had the instructions. So the exons are the pieces in the gene that actually code for the protein that actually have instructions in it. And we call them exons. Think exons for expressed. Like these are the regions that eventually will be expressed that we're going to use to make a protein. But the introns, those are like the in-between things that need to be cut out. And so once we make the mRNA, you're going to have all of the instructions, but broken with I'm not broken, but within the instructions, you're going to have these random pieces that technically aren't part of the gene. And so once we make the mRNA, in eukaryotes, we'll cut out those introns. And we call that RNA splicing. RNA splicing is removing those introns and then joining together the exons. So now you have a nice little set of instructions here without those random things in the way. Prokaryotes don't have introns in their genes, which is convenient. Like if they have a gene, it's like perfect. Like it's like the whole set of instructions. There's no random gibberish within the gene. But in eukaryotes, in our genes, there's always these introns within the gene that need to be spliced out once you make your mRNA. So that's what this picture is showing you here. You might be wondering right now, like, why in eukaryotes do we have these introns? They actually, there's some reasons for it, and we'll talk a little bit about it later on, not in this video. But just for now, know that this is a thing. So this is how we're going to modify the mRNA. We're going to add a GTP cap on the 5' end, a poly A tail on the 3' end, and we're going to splice out the introns. And now you have what's called mature mRNA. This mRNA is ready to go. And so this mRNA can now go leave the nucleus and go find a ribosome. And we can now do the next step, which is translation. Okay, so in translation, the mRNA is going to find a ribosome, and the ribosome is going to take that mRNA strand and read, basically it's going to end up reading every 3 nucleotides on the mRNA. So every 3 letters on the mRNA is going to be read by the ribosome, and every 3 letters on the mRNA we call a codon. And a codon, so those 3 letters, each 3 letters codes for a specific amino acid. And so as the ribosome reads 3 letters on the mRNA, there's a specific amino acid that codes for, and then the next 3 letters, there's another amino acid that codes for, and that's how the ribosome is going to know what amino acids to put together to start building the protein. And so luckily, the scientists cracked the code, and they figured out what every 3 letters, what every codon, what combination of 3 letters on the mRNA, what amino acid it codes for. They figured it out. And so there's something called a codon chart, which shows you, like this picture here, for example, it shows you every codon that's possible. There's 64 possible combinations you could have. So if you have 4 different letters, A's, G's, C's, and U's, and you have a set of 3 of them, there's actually 64 combinations you can make as shown on here. And it shows you what amino acid every 3 letters, every codon codes for in living things. And this genetic code is pretty universal, meaning that every organism follows the same code basically. Every codon in any organism, in us, in plants, in prokaryotic bacteria, and even in viruses, every codon, every combination of 3 letters codes for the same combination of amino acids, which makes this pretty cool. It's a very unifying characteristic of life on this planet that all of our mRNA, when it's read in sets of 3, has the same code in terms of what amino acids it's instructing for. And so you can see those different amino acids listed on here. There's like 20 different amino acids, and this is what each of those codons code for. You guys can see that some of those codons code for the same amino acid. So we say that the genetic code is sometimes redundant, meaning like the same amino acid could be coded for by multiple different codons, each leading. So if it's UCU, that leads to serine, but UCC also leads to the amino acid serine. It's kind of redundant, but it's not ambiguous. And when I say not ambiguous, I mean that if I give you 3 letters, a codon, there is a specific amino acid it codes for. Like, it's not up to debate. Like, if I give you the codon UUU, it will always code for phenylalanine. Like, UUU codes for that amino acid, phenylalanine. Always. It's not ambiguous. It's not up for debate. Like, that 3 letters codes for that. But sometimes there's multiple sets of 3 letters that code for the same amino acid. So we say that it's not ambiguous. It's very clear what every 3 letters codes for. But sometimes there is a redundancy in terms of some sets of 3 letters coding for the same amino acid. So in translation, though, here's what's going to happen. So here's our ribosome, and the ribosome is going to receive that mRNA, and it's going to read the mRNA from the 5' end to the 3' end. So this is very, very, very important that you guys know that when the ribosome receives this mRNA, it's going to read that mRNA in a specific direction, and it's always going to be from the 5' end towards the 3' end. So you can see this ribosome is making its way towards the 3' end as this mRNA moves this way, and it's going to read every 3 letters. Now, this is always going to start with a specific codon called a start codon. So when the ribosome gets the mRNA, it's going to start reading it on the 5' end, and it's going to read it until it finds this combination of letters right here, AUG. AUG is the start codon. That will be the first official codon for this gene. And so if you look here on the codon chart, either this one or the abbreviated one, AUG, that's the start codon, which also codes for the amino acid methionine, or MET. But that's the start codon. That's where translation is going to start. So once this ribosome gets the mRNA, it's going to find the start codon, and then starting with the start codon, every 3 letters it's going to read, and every 3 letters is going to code for a specific amino acid. And the first one's going to be a methionine, because that's the start codon, the first one. And it's going to read towards the 3' end, continuing on. So the way this works during translation is obviously there's a ribosome and there's this mRNA, but then there's also these tRNA molecules. tRNA stands for transfer RNA, and these are special RNA molecules that will bring the amino acids to the ribosome so that they can start being linked together to build our polypeptide, our protein. And so these tRNA molecules have what's called an anticodon. They have a special set of 3 nucleotides called an anticodon that can bind complementary to a codon. So these anticodons can bind complementary to a codon, just like what you see right here. And that tRNA with a specific anticodon is bringing a specific amino acid. Now, the specific amino acid that's being brought by a tRNA molecule, you could figure that out by looking at the codon on a codon chart. So you could look up the codon on a codon chart to figure out what amino acid is it bringing. Be careful to not look up the anticodon. So there is an anticodon that is present on the tRNA molecule, and that anticodon will bind specifically and complementary to this codon, but that doesn't tell you what amino acid is. If you want to know what the amino acid is, you're going to look at a codon chart, which is called a codon chart, and you're going to look up the codon. So if I want to know, okay, this amino acid, this next codon here says UUU, which you can see this tRNA molecule is coming with the anticodon AAA. That's the anticodon, and it's bringing a specific amino acid. If I want to know what amino acid it's bringing to the ribosome, I would look up UUU on a codon chart. And when you look up UUU, it codes for phenylalanine, which is this amino acid being brought here. So don't look up the anticodon on a codon chart. Look up the codon, okay? But know that there is an anticodon that's on the tRNA molecule that's going to cause that specific amino acid to be brought when it pairs with this codon. And so basically they come and bring their amino acid, and then those amino acids get latched together, and then those tRNAs go away. And then the next one comes and brings the next amino acid. And so on during this process of translation, you guys can see those tRNA molecules in this picture coming, bringing amino acid, and then they're going to be joined to this growing chain of amino acids, this polypeptide, and then they're going to leave. And then the next one will come as we read every 3 letters, every codon on the mRNA in the 5' to 3' direction. Really important that you're going this way on the mRNA. And I'll show you an example of why that's important in a second. And so that's going to keep happening, and we're going to keep reading and putting together these amino acids with these tRNAs, bringing each one until we get to a special codon called a stop codon. So once there's 3 letters, there's some codons that actually code for stopping translation, and there's 3 different ones. It's either UAG, UAA, or UGA. If it's one of those codons, that codes for stop. And that's gonna, once the ribosome reads the stop codon, it's going to actually release the polypeptide and disassemble this. So then the mRNA will be released and the ribosome will be disassembled and the polypeptide will be free to go. So that means it's done doing translation. So it's gonna start with a start codon and then read every 3 letters and put together these amino acids with the help of these transfer RNAs. And then at the end, once it gets to a stop codon, the whole thing will then be done. And you have now successfully expressed that gene and synthesized that protein after first transcribing it into mRNA during translation in the nucleus, and then sending it to a ribosome, and then the ribosome reading that mRNA and then putting together with the help of tRNA these amino acids to build the protein, which will now fold up into its final 3-dimensional structure and have a specific function inside the cell and now do whatever that protein's supposed to be doing. And so the last thing on here is just going over some important differences between prokaryotes and eukaryotes during protein synthesis, which I've kind of mentioned a little bit. So first of all, in prokaryotes, prokaryotic cells, they don't have a nucleus. And so their DNA is just floating around in the cytoplasm because there is no nucleus. And so they can actually do transcription and translation at the same time. So we say that in prokaryotes, transcription and translation is coupled. So as the mRNA is being made in transcription, the ribosomes don't have to wait for transcription to be done before they start translating it. So the ribosomes can actually start reading the mRNA and start building the protein, putting together the amino acids while transcription is still happening. Like you can do both at the same time. Now in eukaryotes, that can't happen. In eukaryotes, there's a nucleus that's literally going to separate the process of transcription from translation. So in the nucleus, transcription is taking place and you're making the mRNA. And on top of that, once you make the mRNA, you have to modify it by adding a GTP cap and a poly A tail, and you have to slice out the introns. There's a lot you got to do. And then you can leave the nucleus and go find ribosomes that can then read the mRNA and do translation and put together the amino acids. So those 2 processes are going to happen separately. So if it was a race between prokaryotes and eukaryotes to express a gene and to synthesize a protein, the prokaryote is definitely going to win, okay, because it can just do it all very quickly. Whereas in eukaryotes, it's a more complicated process where you have to do transcription, then you have to like make some changes and modify your mRNA, then you have to go send it and find a ribosome. So a little bit messier or more technical in eukaryotes. Okay, and the very last thing I want to do is show you guys an example where you're going to do, where a question asks you to take a sequence in the DNA and to transcribe and translate it. So on my whiteboard here, I have a random sequence of DNA here. And let's say a question asks you to determine the amino acids that this would code for. So that's going to involve you taking this DNA and first transcribing it into mRNA and then translating it into a polypeptide or a sequence of amino acids. So if you look carefully, though, at the information I gave you, I'm telling you that this is specifically the coding strand in DNA. And so DNA is double-stranded, right? There's a strand, and then there's going to be another strand that's complementary and anti-parallel to this. Now, the coding strand is actually not the strand that RNA polymerase is going to use for transcription. So what you could do here is you could first figure out what the non-coding strand is. The non-coding strand would actually be the template strand. If you remember, the coding strand is actually the non-template strand, but the non-coding strand is the template strand. So just for practice sake, I'm going to go ahead and figure out the non-coding strand. So for the non-coding strand, which is actually the template strand, and I'll put that in parentheses, this is the one that RNA polymerase will actually use. The sequence would be, well, first of all, it would be anti-parallel, so it should be the opposite direction because DNA is anti-parallel. And then if there's a T, there would be an A and a T, G goes with C, C goes with G, and so on. And so if I carefully try not to mess this up, that would be the other strand of DNA in the opposite direction, but complementary, right? So there's my DNA, it's double-stranded. Now, if I want to do transcription, RNA polymerase is the enzyme who actually do transcription and read this information in the DNA and make mRNA. And like I said, it's going to use the template strand, which is this bottom strand, and it's going to build the mRNA complementary to the template strand and anti-parallel to the template strand. Now, specifically, RNA polymerase will read this strand in the 3' to 5' direction as it builds the mRNA in the 5' to 3' direction, which, I mean, I'll put an arrow here to kind of show, like, this is the direction we would be reading this bottom strand, the template strand, that RNA polymerase would be reading it. You don't really have to do it that way. Like, you could just, as long as you keep track of the ends, like, you know, I mean, I could, well, let's just pretend I'm RNA polymerase. So if I'm starting on the 3' end, I'm going to start building the mRNA starting on the 5' end and anti-parallel. So anti-parallel and complementary to this template strand. So if there's a G here, I'm going to put a C. And then if there's a C, I'm going to put a G. And if there's an A, I'm not going to put a T, I'm going to put a U, because U substitutes for thymine. I mean, for U substitutes for T and RNA. There's no U, there's no T's, there's U's. A goes with T, but T goes with A, and so on. And so if I was a RNA polymerase, I would be building it this way, 5' to the 3' direction, as I read the template strand this way, according to how this is set up. But it doesn't really, well, you guys will see in the end, it doesn't really matter as long as you keep track of the ends. So like, where was I? G goes with C, C goes with G, and so on. Let's try not to mess this up. And then A, U, and then this would be the 3' end. Right, so I built this going this way if I was RNA polymerase. As you can tell though, you guys, you could just, I mean, it doesn't really matter. As long as you know it's going to be complementary and anti-parallel, so as long as you just write it complementary to the template strand and anti-parallel, you're fine. Like, I could have done this left to right or right to left. I get this answer here. So here's the mRNA. That would be anti-parallel and complementary to this template strand of the DNA. And this is what's going to be sent to the ribosome. So then the ribosome is going to take this information and it's going to, sorry, I'm just double checking for mistakes. Hopefully that's right. It's going to then read this mRNA and put together the amino acids during translation. And so for this part though, it becomes really important, you guys, that you read this the correct way. So a ribosome will read the mRNA from the 5' end to the 3' end. You need to remember that. Always from the 5' to 3' end, which some of you guys might be tempted to automatically just go left to right. And so you would look up UUA and figure out, okay, that would be the first codon that you would look at, and then the next codon and the next codon, and you could use a codon chart to start figuring out all those amino acids. But you'd actually be doing it wrong because this is the 3' end. This is why the ends matter. You got to be very careful. So technically on this question for the mRNA, I'm going to start on the 5' end, which is over here, and I'm going to read it backwards. So I'm going to read it CGU. That would be the first codon that I would read, the first 3 letters, and I would be going this way, 5' to 3', which it's not always going to be left to right or right to left. And this, it just depends where the 5' end is. You always have to start on the 5' end, which if it was over here, then you would read it left to right like normal. But since it's over here, you're going to read it this way. So the first codon you're going to look up is CGU. And so when I look up CGU on a codon chart, since I have one in front of me, what is it? I can't find these. CGU. CGU is arginine, ARG. That would be the first amino acid. So the first amino acid is arginine. So when I write the amino acids here, you're going to write it in the order that they were put together. So the first one is going to be arginine for CGU. That's the first one. The next one is going to be ACG. So you're going to look up ACG. ACG codes for THR. And then you're going to look up the next one, which is CGA. When you look up CGA, you get arginine again, ARG. And then the last one is AUU. If you look up AUU, that's ILE. That would be the 4th amino acid. And so you're writing the amino acids in the correct order. This is the order that they were put together. Because of the way these codons are written, it should be read 1, 2, 3, 4, going right to left. And so that's going to be your first, second, third, and 4th amino acid. So that's why you guys have to be very careful, and that's why I was stressing about the ends. Now, if you guys go back up here real quick, just to finish this off, I gave you the coding strand You technically could have just started with the coding strand, and you don't really need to write the non-coding strand. I mean, you can, because sometimes you're just used to doing that and making the mRNA complementary to this. But really, I know that the mRNA is going to have the same code as the coding strand. So I know my mRNA is going to be exactly the same as the coding strand, except for the T's are going to be U's, because there's no T's in your in RNA. So right here it says T, T, A, A, G, C, G, C. Well, on here it's going to say U, U, A, A, G, C, G, C, and so on. It's actually the same code, except the T's are now replaced with U's, and it's the same direction, 3' to 5', 3' to 5'. My mRNA is basically identical to that. So I could have skipped this part here where I did the non-coding strand, and I could have just, if I was lazy but smart, I could have just copied this as my mRNA, because I know that's how it's going to end up. But keep track of your ends, because it would be the same direction, the same exact as your coding strand. You would still have to start on the 5' end, and you'd end up with this answer, but by skipping this step right there. And so we'll practice this a lot more in class, but there's a good example for you guys. Anyway, that is it. I'll see you guys later.